right now we've got Maruan Al-Shedivat, who's currently the Director of Machine Learning at Genesis Therapeutics, working on AI and biotech. Thanks for speaking, Maruan. Take it over.
Thank you, Willie. And first of all, I want to thank the organizers for inviting me to cast the workshop. It's great to see a lot of friends, former lab mates. I'll try to be short to not stand between you and the break. I'll talk a little bit about AI and drug discovery and what we do at Genesis Therapeutics, where I work primarily on machine learning. So my talk is going to be split into 3 sections. In section 1, I'll talk a bit about our company. A friend of mine from CMU mentioned that what they know about Genesis Therapeutics is just a company where Maruan works. So I'll try to give you a bit more than that. Talk a bit more about what we do and who we are. And the second part, I'll talk about the platform that we're building internally. And unfortunately, compared to a lot of research talks before me, I have to be a bit succinct on the details because a lot of this is proprietary. So it'll be a bit of a high-level overview. So hopefully, I will not disappoint you too much, but give you enough taste of the problems that we work on. And I'll be happy to talk more offline. And finally, I'll conclude with some research challenges and opportunities that we see in this space. Drug discovery is a very complex problem. It's not just a single kind of benchmark or task that we can solve with machine learning. There are multiple parts in a pipeline. I'll try to tell you what are the most challenging aspects of it. Also, first of all, Genesis Therapeutics is a vertically integrated AI plus biotechnology company. And by vertically integrated, we mean that the goal of our company is to really start from an idea of a therapy for a disease and go through all the stages of drug development, bring eventually a drug to the market, and try to go through this timeline as quickly as possible. To do that, we try to build a very interdisciplinary team of top AI experts, software engineers, and proven drug hunters with a lot of expertise. So our company reflects that, and our team is split roughly into 50-50 AI R&D team, which is based in the South Bay Area, and also a biochemistry team, which is based in San Diego. We also work with a large network of CROs, or contract research organizations around the world, that help us run a lot of chemistry and biology experiments, curating data. Our approach is building this vertically integrated company. And I think there are a lot of companies in this space that try to approach the problem slightly differently. So they try to build AI companies for biotechnology where AI is used essentially as a service for one of the components. And I think that puts them in a bit of a disadvantage compared to us, because some of the collaboration that happens internally at our company is just not possible when they try to sell AI as a service to other companies and rely on that. As given by some of the internal and external points of validation on the business side, we managed to secure multiple partnerships and important deals, such as the Genentech deal in early November 2020, just a year and a bit after the inception of the company, which was a spinoff from Stanford. And then later that year, we raised a pretty massive Series A and generated a lot of interest from the investors. More recently, in April this year, we signed a deal with Lilly. So we have a partnership with them. We're working multiple drug programs and where they use our technology. So apart from these partnerships, we'll also work on multiple programs internally. So essentially, we're working on drugs that we will fully own as a company. And just to give you an example, our platform that we use allowed us to get literally within 5 months to a very promising lead compound. And I'll talk a bit more what that means in a second, which typically takes a couple of years in the industry. So perhaps this is the last slide about the company, which I'll move on to what we are doing in terms of our technology. But we're a small company, so we have to be very focused. We're laser focused on a very specific problem in drug discovery. So our goal is really to enable access to novel chemical space that will allow us to unlock the highest value medicines that we can design for patients. And on the technology side of things, right, we focus on 3D graph AI technology, which I'll talk in a bit. We strictly focus on so-called rational or structure-based drug design, which means that we try to look at the structures of the proteins or enzymes as targets, and we try to figure out how to drug them by looking at the interactions and predicting what kind of interactions that our medicines would make with these targets. And finally, we try to innovate also on the organization building level. As I mentioned, we have this multidisciplinary team where we're building processes internally that allow us to efficiently collaborate. So in terms of the strategy of the company itself, we try to take minimal biological risk. We work on diseases which mechanisms we already know. And the mechanisms could be known for decades. But the problem is that drugging the corresponding targets has been notoriously hard. And we believe that with the technology that we have today, with the compute and a lot of advances in AI, we will be able to crack these problems. We also are trying to bring as much things in-house as possible, because we believe that this collaboration that we're able to establish within our team is paramount for successful drug discovery. And finally, we select very strategic alliances with companies that we believe can help us validate our platform. All right, so I'm going to talk now a little bit about what the AI platform is and what's, how we view what are the steps that we're taking. But before I go into that, let's start with Drug Discovery 101. For many of you, I believe drug discovery could be a topic that you're hearing for the first time. So what is a drug? That's the first question. A drug is a medicine or any substance which has some physiological effect or therapeutic effect when it is ingested one way or the other or introduced into the body. So essentially any chemical compound can be a drug. And how do drugs work? Well, drugs elicit these effects, these therapeutic effects, by essentially fitting their shapes into the three-dimensional structure of the protein or enzyme targets, which you can see an example on the right. So the gray area, the shaded area, is this large macromolecule, which is a protein, and then the colored molecule, small molecule, is a drug that fits into this pocket nicely and essentially locks itself there. And by locking in there, you can use the analogy of key in a lock, right? You insert a key in the lock, some keys would fit, other keys would not. And keys that fit could enable or disable the activity of that. So by enabling or disabling the activity, they will disturb a certain biological pathway and, for example, suppress growth of tumor. This is very high level, but the next question we can ask is why and how do drugs bind to targets? Right, and protein plus ligand, where ligand is a technical term for small molecule that binds to protein. So this binding process is very complex, but essentially it boils down to figuring out a configuration in the state space that is energetically favorable. So in other words, the system converges to some local optimum and stays there for a long enough time if the energy is low. So essentially, we're looking for small molecules that would be able to interact with the protein such that final state of the physical system is going to be low energy. Right? And in general, binding is a very complex process, and simulating this process is extremely non-trivial. It requires a lot of computation, and it's very expensive, even for a single molecule. Now, the problem is that the universe of drug-like small molecules is extremely vast. And it's unclear how do you even approach this problem, right? If you want to do structure-based drug design, at the same time, you want to be able to enumerate somehow and search through the space. And there are more than 3,000 undrugged known targets with validated biology. And we don't have the right tools as of now to be able to solve all of them. So the standard methods known as high throughput screening typically try in parallel a lot of different molecules. So people synthesize them and then they run them through experiments and then they look at experimental binding data. Unfortunately, as you can see, it's orders of magnitude smaller than the possible space, and it's unclear which drugs to select through the screening in order to be able to identify some drugs that would be turned into viable drugs. The other disadvantage is that it doesn't use any structural information. So that's currently one of the industry standards. And our goal is to move away from now the availability of structures. Well, as I said, that we need a structure to be able to simulate in the first place the binding process, even though it is expensive. And the availability of the structures is growing over time. So there are so-called protein database structures that are available publicly, and there are a lot of new EM structures that are being added over time, and the number is exploding with AlphaFold. Now, we technically get a structure for any protein that will be roughly accurate. Some of the proteins of interest, the structures that AlphaFold outputs, are not as accurate in some of the parts as we want it to be, but still, it's a great advancement. And we are coming to a situation where we do have access to a lot of the structures. We could potentially enable this structure-based or structure-based drug development. But the question is, how do we leverage these 3D structures, and especially how do we find the small molecules, given that the computational problem is so complex. Now, AI is a pretty wide area and a pretty wide umbrella, right? And we can get inspiration from the advancements in AI in vision and language. These areas have been already revolutionized by deep learning and new neural network architectures, and the synergy between massive amounts of data sets that were available, as well as the important advancements in the architecture design. Right? And as well as the advancements in massive retraining. So these are the two pillars of modern machine learning that are driving progress in these areas. The question, though, is how do we actually take those advancements and adapt to this complex scenario where we have biological systems, we have simulators, we have access to some data. Experimental data is extremely expensive and hard to acquire. So what do we do in this case? And the approach that we take in Genesis is driven by 4 pillars. Right. So the first pillar is molecular generation. Right. So the first step in drug discovery is that is required in order to pick the molecules. Right. We need to be able to enumerate space. We need to be able to generate molecules that we can pick potential drug candidates from. And to do that, we use controllable and scalable molecular generation techniques, right? Where essentially we start with some initial molecules, and then we allow our system to generate new variants and variations that essentially explores the part of chemical space of interest. And I mean, by controllable, I mean that there are multiple parameters that we have to incorporate, because certain molecules cannot be synthesized, or synthesis is very complex. A lot of logistical questions around that. And all of this needs to be taken into consideration. The second pillar is dynamic potential net. This is our scoring and ranking method used at Genesis. So it's a graphic neural network that can operate on different input modalities. One type of the input modality is the slide, which is a planar 2D graph, right? But we moved away since the inception of the company from that modality more towards 3D graphs and also oral. Finally, the last 2 components are the ML and P platform. right? MD stands for molecular dynamics or molecular simulation, where we actually look at the molecules and simulate how they interact with proteins. We have different simulators that have different computational requirements. So some of them are more expensive to run, some of them are less expensive. And we use that data and to feed back into now.
Good question, Maruan. When you say physics-based deep learning engine, is this merely valid physical confirmations of the molecule, or is there more to it than just confirmation, which is like angles and positions?
So by physics-based, I mean that we incorporate physics-based kind of priors or information into the model itself. We do look at the confirmations of points and as well as sort of their dynamics over time. So if you look at a single confirmation, it's essentially a snapshot, right, picture of the movie, right? But what we are moving towards is analysis of full movie interaction.
Now that's an interesting question because motion is also covered in the molecular dynamics component. So where do you differentiate, if overfitting is a concern, right? Where do you differentiate between what motions go into your upper left model versus what motions go into the upper right? Is it based on granularity, like we clearly showed at the level of like alpha folds and beta sheets up there? So I'm wondering if that's part of it.
That's a great question. So, and not only overfitting is a concern, but also the simulation itself is a concern because it's computationally expensive. So most of that motion goes back into a bright where we simulate and we collect a lot of data, and then essentially we try to distill this information into a suite of models that we define at different speeds and scales. It allows us to, use this at scale.
I see. So does that mean you even consider like not just the full MD simulation, but maybe even coarser models like the ball and spring types of models?
Absolutely, yes. We are looking at the coarser approximation as well as on the other side of the spectrum, we're looking at quantum mechanical simulations.
All the way down to that level.
And finally, we have a data engine that we're building internally. So essentially, we're moving towards this flywheel, which is probably Tesla has built, right? Where you have a fleet of cars that goes out in the world, it collects a lot of data, and feed back in, and iterate, right? So the components that we're focused on, right, is this 3D structure-based AI. So all this data has to be collected from simulations as well as real-world experiments. Use them together. We use a lot of unsupervised learning in order to train with generative and discriminative models. And we also have active learning and vision optimization, which allow us to figure out which data to actually read. Right? We have a lab, we run experiments. So our platform is built to learn from, as I mentioned, multiple levels of granularity of the data. Right? So, and as a first step, we started with pre-training. So we're essentially taking the recipe that has been so successful in natural language, computer vision, pre-train, and then fine-tune the model, and we apply that with a lot of asterisks here to drug discovery. Right, so we pre-train on massive amounts of data sets. We use a lot of rich data sets for that are available for certain proteins that are well studied and used by other companies, available publicly or proprietary databases. Also use a lot of extra data such as ADME data, toxicity, and other different molecules, as well as QM data and data from other types of simulations that we run. And we feed that into our dynamic dimensional net that analyzes the 3D structure, that analyzes the protein motion. And essentially what we're after is learning representations, not just of molecules and proteins, but really molecular interactions between the ligands and proteins. So, and finally, we apply this BI to novel targets. So the targets that we're working on are scarce. There hasn't been, many of them are first, many of the drugs that we are designing, we're aiming to design are going to be first in class, which means that there is no known drugs on the market. And therefore, that have to rely also on some type of transfer learning, right, to be able to transfer data for proteins. The final component of the platform is this human AI collaborative approach that we're taking within the company. And this slide demonstrates real data and real progress over time from March 2021 to roughly end of 2021 and early 2022, where we're able to go through the cycle of molecular generation and then scoring, ranking, and then generating new ideas and then closing this loop with a human in between, right? And this human are actually chemists and biologists that in the lab that sort of orchestrates and participate in every part of the process. So as a result, we're able to start with a pretty rough so-called scaffold or part of the chemical space and refine it all the way to some drug candidates that are extremely potent. And all that was done in pretty short time span. As I mentioned, 5 months ago. All right, so just to conclude, I wanted to mention research challenges and opportunities that I think are extremely important in drug discovery. So the first one is the fact that drug design is a very multi-parameter optimization problem. There are a lot of very strict requirements of what drug candidates can have to be safe, they have to be non-toxic, they have to be effective. And this results in a very massive multi-parameter search problem, which means that essentially there could be tens of different these characteristics that we're trying to optimize at the same time, right? That's extremely challenging. You can try to kind of explore part of the chemical space, which may have high efficacy or high potency of the drug, but at the same time, it may not be safe, right? And we're trying to have to design molecular generation and ranking methods with very strict requirements in precision and recall of these methods. The second one is that there's strong need for out-of-distribution generalization. By definition, I want to design novel drugs. Novel drugs can't be very similar to drugs that are available in the training data that we have. So we have to kind of bootstrap ourselves out from this and make sure that our models are reliable enough when they're trying to make predictions further away from the training data set. Third one is the methods have to scale to billions of compounds that we screen routinely on a weekly basis. And accurate simulation of even a single molecule interacting with a single protein requires a lot of compute. So we won't be able to scale the most accurate and precise methods to the entirety of molecules. So I have to develop methods that are multi-tier and trade off sort of elastically accuracy and compute. And finally, there are some real logistical challenges when we're trying to select and synthesize drugs. You might be surprised by supply chain issues that affect us and our company. Building block availability of different chemical compounds have had a very tangible impact on the drug discovery timeline of our company as well as other companies. And our methods have to take that into consideration, right? Both molecular generation platform as well as the way that we score and select molecules at different stages of the drug program. So all of these are interesting questions, and interesting to see how we can address those by developing methods. Okay, so with that, I wanted to thank you for your attention.